Bismillahirrahmanirrahim. The topic of our discussion is diagnosis of SARS-CoV-2, a virus that causes a COVID-19 disease. So before going into the detail of the diagnostic procedure, just to give you an overview that what does these terminologies SARS-CoV-2 and COVID-19 stands for. COVID-19 is basically the abbreviation of coronavirus disease 2019, and this name is put forth because this disease was primarily erupted in the China during December 2019, and the viral particle belonged to the corona family, so that's why the name of the disease was put forth as the COVID-19. While SARS-CoV-2 stands for Severe Acute Respiratory Syndrome Coronavirus 2. So SARS-CoV-2 is the virus which causes this disease, coronavirus or COVID-19. The major symptoms are the range or the spectrum of the symptom that is associated with these disorders include high fever, muscle pain, headache, sore throat, flu, dry cough, loss of smell and taste, diarrhea, shortness of the breath, and the nasal congestion. So these are the spectrum of the different signs and symptoms. So it is not necessary that all these signs and symptoms should be present in a single patient. It may appear at once. So it depends upon the genetic makeup of the individual. Some of the patient experiences the different type of the phenotype, different type of the symptoms. For example, some patient will experience the high fever and the muscle pain and the dry cough, while some of the patient will experience only the high fever, runny nose, dry cough, loss of taste and smell. So the different patient, they exhibit the different sign and the symptom. And although we also know that the general runny nose or the rhinitis or the flu, they also exhibit the same phenotype, but the difference in the general symptoms and in the COVID-19 symptoms is that the patient suffering from the COVID-19 disease experience a severe phenotype or severe symptoms of all these phenotypes. Structure of the SARS-CoV-2 virus, it's a enveloped virus, circular in the shape, and the membrane is composed of course, the lipid bilayer. Within the membrane, there are present different types of the proteins. Some of the proteins, they act as a receptor for the entrance. So within this envelope, for example, spike glycoprotein envelope membrane protein, while within this envelope is present the genetic material of the SARS-CoV-2 virus, the coronavirus. Coronavirus is basically an RNA-based virus, and this genome is then associated with the nucleoprotein particles which protect this genome. The mechanism of infection of the coronavirus is pretty much distinct. It basically interacts with the ACE2 receptor, particularly the spike proteins. So when the spike proteins, they interact with the ACE2 receptor, this ACE2 receptor, it normally appears on the cell membrane or cell surface of the human body, particularly in the lungs, the liver, the intestine, etc. So once the SARS-CoV-2 particle virus, the spike protein interacts with this ACE2 receptor, it gets internalized into the cell by the receptor-mediated endocytosis, and then it bursts inside the cytoplasm of the cell. So as I said that its genome is RNA-based and it's a positive RNA-based viral particles. So it means that they don't need replication and the transcription. So the genome particle is right away available for the translation purpose. So once the genome is exposed inside the cytoplasm, so within the cytoplasm it uses the host machinery for the translation and translate all those necessary genes that is required for producing the viral RNA particles, its propagation and its spread, and the vital protein particles so that it may take the command of the cell. So once it produces the different type of the protein particles, it then assembles within the endoplasmic reticulum. So within the endoplasmic reticulum, once all these viral particles get assembled, so then it is packed inside the Golgi bodies. So once it is packed within the Golgi bodies, that it moves outward toward the cell membrane. So when it fuses these vesicles, it fuses with the cell membrane, it releases the viral particle to the exocytosis process. And then the released viral particle then may attack on the other cell. And the same process is regulating on, the same process is going on again and again. It's the same mechanism. So the viral particle, by using its spike protein, it interacts with the ACE2 receptor, it gets internalized into the cell. Then, due to the presence of the positive RNA-based genome, the genome is available right away for the translation. It produces the protein of its own type. These proteins then move towards the endoplasmic reticulum. There, the assembly occurs. The packaging occurs in the Golgi bodies. Then the vesicles, it budded off from the Golgi bodies and it moves towards the cell membrane, so where it is released into the space of the cells, and then the viral particle is available to attack on the other cells. So now coming to the actual topic of the diagnosis of the SARS-CoV-2. So there are different methods available in the market in the diagnostic laboratories that are used for the diagnosis of this disease. For example, lateral flow immunoassay, ELISA, RT-PCR, nanopore, targeted sequencing, and the biosensor techniques. So in our lecture, in our presentation, we will mainly focus on these three techniques: lateral flow immunoassay and the ELISA and the RT-PCR. These two techniques, they work on the blood samples, while this RT-PCR is work on the nasopharyngeal sample or the oropharyngeal samples. So in the lateral flow immunoassay, as I said that it only works on the blood samples, particularly on the serum samples. And this process is performed on a small cassette, on a small strip. It consists of three portions or three segments: the sample pad, the nitrocellulose membrane section, and the absorption pad. So the sample is placed on this sample pad, and this absorption pad, it sucks the sample along the length of this nitrocellulose membrane. So different segments, different parts of this nitrocellulose membrane, along with this conjugation pad, it is coated with different types of the antibodies. For example, this M lane or M line, this is coated with the anti-human IgM antibody. That's why it is called as the M line, because it detects the IgM antibodies if it is raised or if it is generated within the patient sample. The G line and the G lane, it is coated with the anti-human IgG antibody and it detects the IgG antibodies, while the control line is embedded or immobilized with the anti-rabbit IgG antibodies. So when the sample is put over here, this absorption pad, it sucks the the blood sample, the serum sample, along the length of this nitrocellulose membrane. So if the patient contains the IgG or the IgM, so along the way it attached over the specific antibodies embedded in their respective line, and they then give us the result. So if the patient did not raise antibodies against the SARS-CoV-2 virus particle, or the other way around, we could say that the patient is negative or the patient is not infected with the viral particle. So in that case, if the antibodies will not be present, they will not be attached over these anti-human antibodies present or embedded along with the M line or the G line. So in that case, we will only get a single band within the area of this control line or the control lane. The other thing, if the patient is infected with the SARS-CoV-2 and the infection is recent, so particularly in case of the recent infection, the patient raised primarily the IgM antibodies. So in that case, we will get two lines, one line in the IgM and the other in the control. So in all the cases, anyway we will get the line in the control line. We will get the glow in the control line, which indicate that the assay is performing perfectly well. While on the other hand, in the test line, there are two test lines, IgM and IgG. So the infection, if the infection is recent, we will get along with the control line, a line or a glow inside the IgM lane. But if some time has passed on the infection and it is not recent, then in that case, along with the glow in the lane of the control, we also get a line in the G, IgG immunoglobulin line. While if the infection is not too recent and not too late, then and the patient has raised both the IgM and IgG, then in that case we will get three lines or three glow line in the lane of IgM, IgG, and a control line. This is the further explanation of that ICT method. For example, if there is no infection at all, then we will only get a line inside the control lane. If the infection is late, is not recent, then along with the control line, we also get line inside the IgG. So it means that IgG antibodies has been raised, are produced by the patient. If the infection is recent, then we will get along with the line inside the control lane, a glow inside the IgM lane. And if the patient has raised both IgM and IgG, then we will get three lines, one in the control lane, one in the IgG, and one in the IgM lane. The next method is the ELISA. ELISA stands for Enzyme Linked Immunosorbent Assay.